about, I've got a couple colors here. The muscle, we're sort of about to get into the physiology of the actual muscle contraction. Well, the muscle stimulation, how a muscle is stimulated by a nerve, and then how it's going to contract. So it's always a good idea with any topic really, but this especially, and this pertains also to lab when you're studying the bones, the muscles. Call it chunking, just to try to break things up into little compartments, into little kind of bite-sized pieces, so it doesn't seem so overwhelming. The process in this chapter we can divide into 3 or 4 really easily identifiable kind of parts in sequence. And we're going to start with, well, we talked already, we're doing some background. We're talking about the anatomy of a muscle down to the micro level, down to the parts of the muscle cell. But the actual, isn't them. Of course, step away. Injured press. Okay. Let's put the ringer on. We're going to break the physiology down into first the excitation of the muscle, how a nerve stimulates a muscle. So you're going to learn the steps in that process, and then there's a little period of time between when the muscle is stimulated by a nerve and it actually starts to contract and generate force. We call that coupling. So we have excitation and contraction. In between them, we call it excitation, contraction, coupling. Like the little connection between them. So that's a little stage. We've already talked about actually a lot of what happens there. And then we have the contraction itself and then relaxation of the muscle once the contraction stops. Did you have a question? Oh, okay. I thought you didn't. All right, so let's, we were talking about, we zoomed in from the whole muscle down to, we have bundles of muscle cells. What were those called? Like inside? Yeah, fascicles. Okay, and fascicles are made up of individual muscle cells or muscle fibers. And then inside the myofiber, the muscle fiber, we have those myofibrils. We wrote this down last time. And then finally, we're down to the myofilaments inside those myofibrils. And they overlap in these individual units called sarcomeres. The functional unit of the cell, of the muscle cell, is called a sarcomere, or the functional unit of contraction. A sarcomere. Hello. So let's draw the parts of a sarcomere. I have these 2 colors. So, and again, these are repeated. There's thousands of them within each muscle cell. So we have this kind of zigzag line that forms the wall of the sarcomere. Let me kind of try to do it accurately here. Line it up as best I can. Tech support? Okay, yeah, so several issues today. First of all, I can't get this computer to turn on, which is what I normally use. It's not turning on. And then I was here with Marvin last time. I bought this adapter. I want to try to use my laptop, but I can't get my laptop to, he got it to work. I don't know what combination of things I need to press to get this to display. You can help me with that. Well, that's a good sign.
Yeah.
That looks like this computer though, but
Yeah.
yeah.
That's this one right here on the back. So,
Oh, so what did you just do to turn that on?
Room PC.
You hit that and turn this on? Normally this is on before I even touch that screen.
Sometimes you go to sleep mode.
Yeah, I was trying everything. Okay.
Okay, so go ahead.
What about, can you get the laptop to display?
Let's see.
It's the HDMI, which is trying. Oh. What's up, you guys? Hello? Yes. Yeah, someone just came in. He's here. Thank you. Thank you.
That's the one.
That's weird. Oh, that's the desktop. All right, while we're trying to get this projected,
Nope.
let's continue. So the sarcomere, functional unit of contraction, we have the walls of the sarcomere here, which are zigzag like that, so it's easy to remember. We call these the Z, we give it the letter Z. We call them Z discs. The Z disc and the Z disc. And why is it a disc? I mean, if you picture the myofibrils are round, tubes, and, the sarcomere is round, so it's kind of, but these walls of the sarcomere, but we see it with the zigzag, which helps remind us that it's Z, okay? Then we have, let's say, attached to the Z disc, let's do it in blue, because I'm going to go with the colors that the book uses. We have the thin filaments. I'm going to try to line them up as best I can, something like that. So we have our thin filaments, which are made of what? What protein makes up the thin filaments? Actin, good, so let's write that. Okay, in blue, actin, or we'll say thin, the thin myofilaments. Thin myofilaments. That says thin. I'm going to write myofilament just to remind us of that term. Sorry, it's not that visible, the blue here. Okay, and then, I'm going to break this in half. We have lined up between the thin filaments, we're going to have the thick filaments. So they're like this. I'm going to draw them a little thicker in here. Like that. And remember, we can draw, they have these little myosin heads, these little hooks on them on either end. Actually, on the bottom too, like that. These are the little, what we call myosin heads sticking out of the, well, let's write that. The red thick filament is myosin. Okay, we say that's the thick filament, thick myofilament.
Professor?
Yeah.
It's also made out of actin, the protein?
Well, actin is the thin filaments.
Uh-huh.
Myosin are the thick filaments.
Oh, okay.
Okay? These are the two contractile proteins, the two protein fibers, filaments, that are going to grab onto one another and pull and shorten the sarcomere. Okay, so one other thing, we have, these aren't just floating in the middle there. They actually are connected by these coiled filaments that kind of come from the Z disc also, these very springy coiled proteins. We just do it like this. Okay, so those, let's write this. These are a different type of protein. These are elastic, and they are called, let's write elastic filaments. And the name we give to that elastic filament is titin, T-I-T-I-N, it's the protein. It is actually the longest protein that we know of. It is super, super long and coiled up so that it's nice and springy, okay? It's something like, remember we talked about proteins made of amino acids? This is close to like 200,000 amino acids. Now that's a thermos right there. 2 hands. Okay, so yeah, I usually do a little thing, I have a video, or, I looked up like, what is titin? And someone said, oh, it's the longest word. Because each amino acid is part of the name of this protein, and it's like, someone decided to type it out, and it's this website you got to scroll through for like 5 minutes to see the whole thing written, because it's 200,000 amino acids long. And some maniac decided to make a YouTube video of him saying the name of it. It's a Russian guy. And it's like a 5-hour video. It's ridiculous. People have too much time in their hands. And there's a lot of views, which is also concerning. So, very, very springy protein, but this is, you should know the name, it is titin. It kind of keeps the thick filament aligned where it's supposed to be, but also when this is going to contract, and these little heads are going to attach to the actin, the myosin heads, pull them in, and then when they release, it will spring back to where it was. It also allows us to stretch the muscle if we're stretching it. Awesome. Okay, so let's name a couple other parts of the sarcomere. We know like what the components are. When we looked at a muscle, remember we looked at a microscope at the muscle, we saw it was striped, like striated, right? It had that sort of striped appearance. And that's because of the overlapping thin and thick filaments. I'm gonna draw another sarcomere next door. It would look like this. So here's like, you know, the other one right here. We're moving, you know, and then there'd be a filament going there. Okay, kind of like that. And we'll draw a little squiggly line. Okay, another sarcomere. I drew like half of the sarcomere over there. Where it's just the thick filament, it's gonna look darker. Okay, where it's the thick filament only is what we call the A band. Whoop, see ya. Okay, the A band. Where it is light, which is from here to here, okay? Like where it's just thin filament, elastic filament, no thick filament, it looks lighter. We call that the I band. And just make a little note so you remember the dark and light bands. The light band is the I band, your eye sees light. Just remember that. So you don't mix those up. There'd be another I band over here, you know, going into the next sarcomere. This would be another I band like that. This little zone in the middle, right here, where it's just thick filament. See how it's like here it's thick and thin? So it's like even dark, and I'm going to show you in a picture in a minute. There'll be a little bit of a lighter section right in the middle of the A band, where it's just thick filament, no thin filament. We call that the H zone. And make a little note, H is like in the halfway, it's halfway, right in the center. The H zone.
What's the A band again?
The A band is basically where the thick filaments are. Okay, it's where the thick filament is. The I band is where it's just thin filament, lighter. And the H zone is right in the middle of the thick filament where you see here it's overlapping, thick and thin, thick and thin. Here it's just thick, right in the center. So this is like the H band right in the center there.
Oh.
So the A band is like interlocking thick and thin, right? For the A?
The A band, yeah, it is both thick and thin, except for the very, very center of it, which is the H zone, we call it. But the whole thing would be called the A band. The H zone is kind of part of the A band, let's say that. It's right in the center of the A band. Okay, so those are letters Z, A, I, H. Okay, just be able to label that diagram and know what the parts. Okay, so this is, you know, if I ask that, it's myosin. If I ask the thin filament, actin, titin, elastic. Okay, these are the parts of the sarcomere. When a muscle contracts, the Z discs get closer together. It's not that the thin and the thick filaments shorten. They just overlap. They pull and overlap more. So these walls move in. The I bands actually get smaller. You see how that would get smaller? The A band's going to stay the same because the thick filament is there, but we're just overlapping with it more. So the I bands get shorter during, yeah. Any luck?
Alright, so it's kind of crazy because we gotta do just click on like an HTML.
Uh-huh. But do you think maybe it's the adapter?
Could be because I went back and forth, okay?
Yeah, it's flickering.
Click it. So,
All right, I don't know. If Marlon's around, he was here the other day and just flicked something and it worked. I don't know.
Just told me that you
Oh.
HDMI, but
Do you want to try? Because I used his adapter the other day. Do you have one there? Maybe we just troubleshoot it. Yeah.
Find him, I'll be right back.
All right, if you don't mind. And then also one last thing. Yeah, take that out. It's asking me, of course, today my Wi-Fi password has to be reset, and I'm putting in. This I closed it because it was kept popping up, but I'm trying to enter my old password. It's not allowing me. It's not allowing me to put in a new password. Any idea?
You have to go password right now?
Yeah, I'm pretty sure. I mean, I'm assuming. Is it the same password I used to log into my computers, or?
It should be, yeah.
That's what I thought. So
That's the one that you use.
yeah, for the one I used to log into a computer, not to the web services. Okay, so that's that one, and then I'm trying to enter a new one. Let me just make a similar one, but
You can.
Oh, it's gotta be very different?
Very different, yeah.
All right.
It's gonna be different.
let me,
You can reverse the order if you want.
it's very confusing in my head. Let me try. Whoops. Hang on, I'm gonna have to write it down, because I'm
Yeah.
gonna forget it. Let me just make one up.
Anyway,
Like I like chorus, but like
I think you can go back and make it.
yeah, but it's so I wanted it to be like yeah. That's the only way.
I hope it was
I'm so
Okay, now let's try that. So account name, that's this, I believe.
and then 3 other people didn't vote. They should have just voted and most of them actually know so would have had alien because it was
Yeah.
unanimous.
yeah.
Leave it like that.
All right. And-
I'll be right back.
Okay,
I'll be right back.
let me see if I can get this one working.
Oh, I'll take it back.
Now this isn't on at all. Ay. Wake up. I had all my clicker questions ready to go today. Nothing's, now it probably won't even let me log in. Let's see if this works. Oh my God. No. Hey, it happens to all of us.
Oh, you have to. Oh, you have to.
Yeah, I know. But I already changed it on there. We'll see. All right, I'll let them go. All right, we're going to try to do this from my memory here. Okay, so here's our sarcomere. Now, actually, I can look at my notes and at least keep myself on track here. Hopefully. Or no. What did you do to my computer? Okay. So, let's, I'll show you a couple pictures to the sarcomere as soon as I can. But now let's talk about a muscle stimulating a nerve, okay? Because ultimately, in order for a muscle to contract under normal circumstances, a nerve is going to have to come, and then we're going to talk about nerves. Nerves are an organ that have individual nerve cells in them called neurons. And a neuron is going to attach to a muscle. So here comes a neuron, and we have a little end. We call that the neuron terminal. And we have a muscle right here, okay? Let's just draw a muscle like that, okay? And it's going to stimulate the muscle. It's going to release a little chemical neurotransmitter onto it that's going to cause the muscle to contract. But let's zoom in and see what's happening here. So we have something called, that little place where the nerve releases the chemical into the muscle is called the NMJ, which stands for the neuromuscular junction, okay? And this is a specific type of synapse. You may have heard that term before. It's a chemical synapse. We can have a synapse between one nerve cell and another nerve cell, a nerve cell and a muscle, a nerve and a gland cell to secrete. That's okay.
Back to what you said, nerves are individual organs and muscles.
Yeah, so, whoops. A nerve, I'm just defining some terms. Let's say this is a nerve. Like, you can see a nerve, you open the body, you can see a nerve, which is like a tube. And a nerve contains lots of little individual nerve cells. It contains thousands of them. Kind of like a muscle contains, many, many thousands of individual muscle cells. So an individual nerve cell is called a neuron. Okay, this is a nerve cell. Okay, now a neuron, we said, comes out, and maybe this one is going to secrete its little chemical neurotransmitter onto a muscle. and it's going to cause that muscle to contract. That's a terrible drawing of a muscle, but you get the idea. Okay, let me zoom in here a little bit. I'm all right. So this right here, where it releases that chemical onto the muscle is called the neuromuscular junction. Okay? Did I answer your question? No? You don't understand that? Okay, let's say this is a muscle. Just humor me. This is a muscle. A nerve actually has to come to that muscle, and a nerve cell is going to release a chemical onto it. If the brain sends an electrical impulse down that nerve, whoop, goes down your spinal cord, comes out, goes to the muscle, it releases a chemical on it, and a whole bunch of steps are going to happen that tell the muscle to contract. Okay, so this is how we communicate to our muscles to contract. And that little synapse, a synapse is where a nerve releases a chemical that stimulates another cell, in this case muscle cells, to do something. Okay, so that is called the neuromuscular junction. And I'm gonna put my art skills to the test here. The neuromuscular junction, I'm gonna blow it up. So here is the end of our nerve. Let's draw it like this. This is the nerve, I'm gonna make it kind of big. And it's got this little bulb at the end of the nerve, at the very, very end of that nerve cell. And some of these nerve cells could be super long. You could have one of these cells that starts in your brainstem, above the foramen magnum, right? Foramen magnum, and goes down your spinal cord, one cell all the way down, comes out, goes through your butt, down your leg, all the way into your foot, and stimulates a muscle in your pinky toe. One cell. Okay? But at the very, very, very end of that cell, we're going to have something called the synaptic. It's sometimes called the synaptic knob, sometimes called the synaptic bulb, because it's kind of rounded like a knob or a bulb. It's also called the synaptic terminal, which makes sense. It's the very end of the act of the neuron. And this part, the long part of a nerve cell, you may have seen this before. Do I have another kid? Oh, I got stuck. There's another blackboard here, but okay. That's not good. Can't even get a blackboard to work today. Jeez. It is one of those days. Okay, you may have seen a picture of a nerve cell like this. You got that star-shaped body, and it's got some receptor endings, little dendrites, and then it has this long process. And that process is called an axon. And that's the part of a neuron that can be super long. The body of the neuron is in your brain, but the axon of the neuron, that long, can be super long. Could be, a meter, 2 meters long. So sometimes we call this the axon terminal. It's just the end of a neuron. It's the very, very end of a neuron. Now, at the end of the neuron, we're going to have inside these little vesicles, these little bubbles. These were made, we're going to talk about this probably next time, but these were made all the way back in the body of the neuron, wherever that was. This neurotransmitter gets stored in here. We have little chemical neurotransmitter. You may have heard of some neurotransmitters. The ones in your brain like dopamine and serotonin and maybe adrenaline is one, epinephrine, acetylcholine. Oh, there's my computer.
All right, so it was your
Oh, it was. I just opened it out of the box. I guess I got
Yeah, okay.
another return to Amazon. And I thought I bought the gold.
What time do you finish the class so we can
10:45.
So I'll be here like 10:40 or so.
Okay, perfect. Ooh, I gotta check something. Hang on. Yeah.
Yeah, that's the only reason I can.
Give me half a second here.
Yeah. It was.
Okay, no, that's not today. Okay. That'd be great. Yeah, you just come at the end. All right, perfect. Okay. So I think I can shut this off and just do the notes. Okay. So guys, I apologize for today being all over the place. What time is it? It's 10. All right, so here's, we're looking at that sarcomere. There's like your kind of drawing with the letters there that we just went over, the thick and the thin filaments, everything's labeled there. Just put it a bunch of different ways. This was just showing that springy large thing, and there's a link to the website where you can see the name of it if you want. It was cool. And I remember my kids coming home from school one time. They gave them like a little dictionary, you know, thesaurus and a dictionary. In the back, it said the longest word in the language, and it was like, I guess they just ran out of things to do in the dictionary. They had like 15 pages of just the name of this this protein, Titan. But you get the idea. Okay, and so different pictures of the sarcomere with them labeled there. You can kind of see it inside of a myofibril, what it would look like. So take some time to look at these. Here's a very simplified drawing, thick and thin filaments, A-band, I-band, H-zone. You can see what it would actually look like here. Here's a bunch of myofibrils, like 5 of them, see? And you can see the dark band, that's the A-band. There's that H-zone right in the middle they showed over here, but that little light zone right in the middle. There's the I-band, the Z-disc. So take a few minutes, make sure you review the parts of the sarcomere. That's showing a relaxed sarcomere and then contracting. You'll notice the I-bands get smaller. When it contracts here, regular, look at the I-bands got smaller when it contracts. See? The lighter part of the smaller. Okay, so now we're going to look at, there's that nerve up there, and one little nerve cell, and it's stimulating a few muscle cells. We actually have that. One neuron can branch multiple times and make, like, let's say 10 muscle cells contract. We call that a motor unit. Okay, we're going to get used to the term motor. Motor is the opposite of sensory. When we talk about the nervous system, all the information coming into our brain is sensory information. So we have sensory nerves, sensory neurons, and then everything going out we call motor. So motor nerves. Now, when a nerve goes out or a nerve cell to a muscle, voluntary, we call that somatic motor. This basically means skeletal muscle, motor. Okay, voluntary skeletal muscle motor. We also are going to learn about something called visceral motor. Does anybody remember the term viscera? What does that kind of mean? Your viscera is another name for your what? Your internal organs. So your visceral motor system is called your autonomic nervous system. Those are nerves that go to your heart, your lungs, your liver, and your, all these things you don't think about voluntarily. But anyway, somatic motor neurons are the neurons that stimulate our skeletal muscles. That's a whole division of our nervous system, whole part of our brain. Frontal lobe is dedicated to that. Okay, but a motor neuron and all the muscle cells or fibers that it innervates meaning stimulates, or supplies with nerve supply, is a motor unit. So we could say, let's say I need to lift this pen. My brain knows from experience, oh, that's pretty light, I've lifted those before. I only need like one motor unit. I just need a few muscle cells to contract and I can flex my bicep to lift that. But I'm gonna lift, this bag, and I know from experience, yeah, I packed a bunch of heavy stuff in there. I'm gonna need like 5 motor units to do that. Or now I gotta lift this thing out of the way. I'm gonna need a whole bunch of motor units. So our brain knows that. That's why you see little kids picking up things, knocking stuff over, learning. Your brain is learning what things weigh and what they are. You ever have that experience? You go to pick up a box, you think it's full or think it's heavy, and it's empty, and woo! Because your brain is going, okay, I need this many motor units to lift the box that big, but it's empty. Yeah.
Does motor unit seem to like decrease as you get older, like when you're like elderly? Because
I wouldn't say your motor units decrease. You still have the stimulation to the muscle. Unless, of course, if you block that motor nerve, if that motor nerve gets cut, gets damaged, your muscle's paralyzed, right? But as you get older, the muscle cells do get smaller, so the response won't be as strong, but you'll still be able to stimulate the muscles.
Okay.
Obviously, unless there's some damage. Okay, there's just an example of a couple of neurons coming through a nerve from your spinal cord, central nervous system. It's showing 2 motor units. This one supplies these couple of muscle fibers. This one supplies these muscle fibers. So they're all throughout the muscle. I could have, if I have 1 motor unit, again, very little contraction, several stronger. We're going to talk about that later, how we control the strength of a muscle contraction. But let's focus, we're going to focus right on the end of one of these little neurons, right on the very tip there, is what we call the neuromuscular junction. Well, I guess I could close this now. No, I can't. Oh, that was weird. I think that just timed it. That's coming from here, right? And now I don't have a cursor. It really is one of those days. Nothing working. Okay, that was working a second ago. Now not so much. Can I still? Unless, is that showing this screen? People are leaving. Okay, I wonder, is that, oh it is. That's weird. So this is showing on there now. Guys, I'm as lost as you are. All right, let's see what we can do here. All right, so here is our neuromuscular junction. Okay, so picture this. This is the end of a long neuron. It's made a long journey. It's come all the way from your brain, your central nervous system somewhere. And here's the very end. So this would be the synaptic knob, this whole thing right here. This is the synaptic knob, the synaptic bulb, the terminal, synaptic terminal, axon terminal, it's the end. And it contains these little vesicles. These are called synaptic vesicles, and they contain a neurotransmitter called acetylcholine. Okay, I'm going to have to erase some of this. Just want to do a little beautiful artwork that I didn't need. Okay, whoa. Do this. So the synaptic vesicles, okay, so when the synaptic knob contains synaptic vesicles, V-E-S-C-I-C-L-E-S, okay, which contain, and I'm going to do the abbreviation that you'll see a lot. A, big C, little h. Acetylcholine. That is the only neurotransmitter that is used to stimulate our skeletal muscles. What's that? Oh, I did spell it wrong. Thank you. V-E-S-I-C-L-E-S. All right? I'll take your word for it. It looks right. Okay. Vesicles contain acetylcholine. That is our neurotransmitter. Okay, a couple other terms. Oh, let's do this because I want to. Just forgot I gotta do this. Oh, maybe not. Okay, hang on. I'm going to switch this to a slideshow, and maybe that'll do it. There, maybe.
I think press the drawing thing first on your
Where?
computer.
On
On the computer.
on here. Yeah, see, it's different with the laptop.
Oh, it's supposed to
Yeah, I got to do it with the mouse. Okay, so this whole thing's the synaptic knob. These are the acetylcholine are inside these synaptic vesicles. Now this is the muscle cell right here. So there's a little gap. That little gap is called the synaptic cleft. That's the little space in there. The space. And this part of the muscle cell, this is the sarcolemma, right? This is the cell membrane of the muscle cell, and it has these folds. What do we think of when we see those folds? Increase surface area. Because all along on there, I'm going to draw them in. Actually, let's do it in green because it's written in green. These are ACH receptors. They're going to be all along here. I'm just drawing little dots. I'm trying to anyway. Okay, I don't know if can you see that? Okay, they're all along here. I'm trying. Those are ACH receptors for the neurotransmitter, right, on the muscle cell. So we can kind of picture what's going to happen here. But those are called junctional folds of this area, this little place on the cell membrane of the muscle where the nerve is sitting is called the motor end plate. It's like where we're receiving the nerve. And then the motor end plate has junctional folds to increase surface area, and those are covered with ACH receptors. Okay, so just to back up, we have a nerve, the synaptic knob or terminal, with containing synaptic vesicles containing acetylcholine. There is a little gap, a little space, a synaptic cleft, and then on the other side of the cleft is the motor end plate of the muscle, of the sarcolemma, with its junctional folds containing acetylcholine receptors. And we kind of guess what's going to happen here. A signal's going to come down this nerve. The brain said, okay, I want to send a signal to that muscle. When it gets to the end here, it's going to cause these vesicles to release into the synaptic cleft, to release acetylcholine. Normally it's sitting inside these vesicles. When the nerve signal gets there, we're going to go step by step. It's going to release that neurotransmitter, acetylcholine. It's going to bind to these receptors. And then what's going to happen? It's going to open up certain channels. It's going to open up sodium channels, potassium channels. We'll get to the specifics. It's going to make the charge change inside that muscle cell. And what that, for shorthand way of saying it is, the electrical signal comes here, causes a chemical to get released, which creates another electrical signal now on the muscle cell. Now we're at that point that we talked about last time. Now the electrical signal is just going to spread in all directions along the muscle cell. Where's it going to go? T-tubules. Remember those? Okay, so we're back. We're just behind that. This is where it starts. Okay, so let's look at another. I'm going to kick that out of the way. Okay, just some things labeled here. You could. Okay, there's the synaptic vesicles with the ACH. Here is the sarcolemma with ACH receptors, the junctional folds, the synaptic cleft. Okay, don't worry about Schwann cells yet, we'll get to that. Okay, so you get the idea. Don't worry about the myelin or the Schwann cell yet. We'll do that in the nervous system. It's just like insulation for the nerve cell. Okay, and then you can see there's the muscle cell. There's there's a myofiber. There's a sarcomere right there. Right? Maybe a nucleus, a mitochondria. But don't worry about that. Okay, so here's one of those times where I had to decide: are we going to teach the muscular system first, or nerve physiology first? We have to understand, because they're both electrical, kind of excitable tissues, cells. They have similar properties that we have to talk about a little bit here. So muscle cells, nerve cells, they have what's called a polarity across their membrane. They have a definitive charge difference that we actually work to create. Remember the sodium-potassium pump you learned about in lab? Right? Remember, what did it do? It pumped in, how many sodiums did it pump out of the cell?
2.
No, sodiums, 3, and 2 potassiums back in. So every time that sodium-potassium pump works, it takes 3 sodiums against the concentration gradient, right? Let's just remember this. There's much more sodium normally outside our cells than in. Sodium is the primary extracellular cation, positive ion. So just picture this, much more sodium outside the cell, and the opposite is true for potassium, K, right? K plus, they're both positively charged cations. But sodium is outside, potassium is inside. So there's a big concentration gradient. So what the sodium-potassium pump does is it pumps out 3 sodiums and brings 2 potassiums back in. So 3 positive charges go out, only 2 come back. What's the inside of the cell going to become? More positive or more negative?
Negative.
Negative, right? I'm getting rid of 3 positives, but only bringing 2 in. So each time it gets a little bit more negative. I'm spitting out 3 positives, but only 2 come back. Okay, so each time we spin that cycle, it's getting more and more negative inside the cell. So we do that in these cells. It's almost like creating like potential energy, because not only does sodium really want to get into that cell down its concentration gradient, and potassium wants to get out, but also now the inside of the cell is negatively charged. That sodium really wants to get in, not just down its concentration gradient, but also its electrical gradient, right? Positive, negative charges want to get together. It's like a magnet. But we got the cell membrane in the middle. And sodium and potassium are water soluble, which means they can't just go right through that cell membrane. They got to go through a channel. but those channels are going to be closed. Okay, normally. But when they get stimulated, they'll open up. Okay, so let's think about that for a second. So we have something called when the muscle cell or a nerve cell, we're talking about a muscle now, is just at rest, nothing's happening. We say it's at resting membrane potential. And it's going to be much more negative inside the cell. You don't need to know exact, but somewhere like negative 70 to negative 90 millivolts, more negative inside the cell than out. Okay? And more negatively charged inside the muscle cells. What's going to happen is when an action potential, which is a nerve impulse, reaches that synapse, causes the release of a neurotransmitter, we said that the neurotransmitter ACH is going to bind to ACH receptors. Those receptors are going to be ion channels. They're going to open in response to that chemical, ligand-gated, we call them cation channels. So, what's going to happen? If I open sodium channels, what's on a muscle cell, what's going to happen to sodium? It's going to rush into the cell. There's much more of it outside than in, concentration gradient now, and it's negatively charged inside, and these positive, they just rush in there super, super fast. And what they actually do is change it from negative, it goes all the way to positive. Really quick. And then right as soon as that happens, the sodium channels close and potassium channels open, and potassium's gonna go out, and it goes right back to negative. It's a quick flip-flop. It looks like a spike on a graph. We're gonna study that spike. An action potential goes from negative, positive, back to negative really quick. But what that does, just picture this and kind of, we'll go into more details of the nervous system, but once one action potential happens on the muscle cell membrane, that first one, it triggers another one in all directions. So there's another action potential next to it, then another one gets triggered. It's like knocking over the first domino when you line up the dominoes to knock over. If the first one happens, it's going to spread across that entire cell. So it triggers another action potential next door, next door, next door, and it just keeps going and spreading. Okay, so spreads all over the cell surface as a nerve signal or an impulse. We call it an AP, an action potential.
Professor, so when is that negative energy is resting, right?
Yeah, that's at rest. It's negatively charged inside the cell. Yeah.
And then if it turns positive, that means it's switched a little bit and it goes back to negative.
Yeah, it's going to switch. It's going to go from negative to positive back to negative. I'm going to show you that in a little bit more detail in a minute.
And then when it's resting, it's more negative side.
Yeah, at rest, it's the inside of a muscle cell is negatively charged. Same thing with a nerve cell we're going to see. And I make the analogy to like a battery, right? You have a battery with a positive and negative end. One side has positive charges, one side has negative charges. There's a wall in the middle inside the battery. They want to get together, but they can't. There's a wall there. But when I attach a wire to both ends or a circuit to both ends, now the negative charges can move, and they move to the positive side through the wire, and that can power whatever's attached to that wire. It can cause a motor to turn or a fan to turn or something. Same kind of thing here. Okay, these are the four little chunks we said we break this chapter down into. We're kind of talking about excitation now. That's with a nerve. That happens at the NMJ, the neuromuscular junction. Then we have that little EC, or excitation contraption coupling, which is that's one of the things going down the T-tubule and making calcium get released and unlocking the stuff we were talking about last time. We're going to bring it all together. And then we finally get the thick filament grabbing onto the thin, contracting, and then we'll talk a bit about relaxation. So you can break it down into these four parts. Okay, this is also important. I want you to know this term. Let's just say the muscle's sitting right here. Okay, there's no tension in the muscle. It's at zero. There should be a zero there. The muscle's just at rest. Even though it's a little bit of our muscles, even though we feel like our muscles are just relaxed, like we're not using them, they still have a little tension in them. There's always a little bit of tension so that they're ready to go. Anyway, so maybe that's why it's not exactly zero here, okay? But let's say we stimulate the muscle right here. We just give it a little, we give it a little shock. Like maybe we hooked an electrode to it, which is similar to a nerve stimulator. Notice that before tension starts to generate, there's a little period of time. That is called the latent period. And I want to just write down, the latent period right down there is the same as that coupling, that second part right here, okay? Because excitation is just what happens right there at the synapse, and then contraction starts right here where it starts to go up. So that little period of time, latent period, the time between stimulation and contraction. And that's what I was telling you, that all this stuff is going to happen, all these steps. The neurotransmitter gets released, it binds to the receptor, sodium goes in, potassium goes out, blah, blah, blah. All that's happening right in there, in that tiny little time frame right there. The latent period, the period between excitation or stimulation and contraction. See, the contraction is rising, tension, and then relaxation coming back down. So just know what we mean by the latent period. And you'll know what's going on. That's what we're going to be talking about. All right.
So the latent period has nothing to do with resting, right?
Well, it's right after. It's sort of like the muscle, to all appearances, is resting still because it hasn't generated tension yet. It's been stimulated, but there's a few things that have to happen before this happens, before the thick and the thin filaments grab onto one another and pull. Okay? Those are all the steps we're going to talk about between excitation right here and contraction.
Question.
Yeah.
Does that affect somebody's quickness or slowness to response? Like when something's coming at you and
Oh, like reflexes? Yeah, I mean, that can certainly be, affected by how long this takes from one person to the next. Yeah, your reaction time. And we'll talk about what could affect that later on. Okay, so here's the first part. Here's our excitations, just another diagram of the end of a nerve. Okay, look at the end of a nerve. We have a little synaptic knob here. And let's start thinking about this in a sequence of events. And you got the numbers here. And I used to write them down, but I'm a little short on time, but they're written on this diagram right here. So let's go through them, and then we'll watch a little video. I'll show you the video of this. So an action potential arrives at the axon terminal. That means a signal has come down the nerve from the brain. We decided, hey, I want to lift up this mouse and see what the heck's wrong with it. Okay, whatever it is. So that's first. Then it gets to the end. It gets to the very end. Here's that action potential. And it's kind of traveling along the surface. I wish they showed it right, because action potentials travel right across the surface, because it's because of ions moving in and out of the cell. Sodium, potassium, flip-flopping. Anyway, when the action potential comes to the very end, we have what are called, let's label these, voltage-gated calcium channels. Here's where we're going to talk about calcium again. Calcium comes into play twice. Here's the first time. The action potential, the change in voltage, that quick flip-flop that's happening, flip-flop, gets to this calcium channel, it opens it. Calcium also has a gradient. There's more calcium outside a cell than in, just like sodium. Calcium's going to go in. Calcium going in makes these vesicles move to the end and open up. What do we call that, by the way, when a vesicle opens up and releases stuff out of a cell? Exocytosis, exactly. That's what happens at every synapse. When a neuron releases its neurotransmitter, it's exocytosis. So in this case, we release these ACH. Calcium entry causes ACH to be released by exocytosis into the synaptic cleft, right? This space right here. Look at it. It goes right across. It binds to ACH receptors. ACH receptors, what's going to happen next is these are going to open up and allow sodium to rush into the muscle cell.
And then potassium goes to
Okay, here's the neurotransmitter. I think there's just like a GIF or whatever, it's just playing. But the neurotransmitter is the orange and blue. Orange and yellow. Okay. Brain falling apart. Okay, and you saw what happened. When it binds, it opened up those little things. There it goes. And it's going to let sodium is in the red there. Pink. Whoop, they open, and now sodium is coming into the muscle cell. Let's watch this. Let me just cue up this video. Okay, let's do, oh,
The one.
yeah, I'm not connected to the internet. Okay. Well, good, at least we have this little video here. But those videos I was showing you in, and I think I linked to them in the Blackboard folder, but also in the Pearson study area. Go to chapter 9 or go to something called, there's two things, if you want to write these down. You go to animations and videos, go to A&P Flicks, F-L-I-X, like a flick, a movie. And you'll see the first three, click on the first thing, the first three are going to be from this chapter. But also, if you go to, in the Pearson study area on Blackboard, go to Interactive Physiology, IP. There's some great tutorials on this that go like step-by-step little videos, and they say, hey, add calcium, see what happens. And you open up the calcium channel and you see the next thing that happens. Maybe it stops and asks you a question. The chapter 9, which if they're not open yet, I'm gonna make sure they're open. There's 2 of them, study modules, practice. I built the videos right into them with follow-up questions, so you guys can do that. Those of you in my CoGen thing, I did have all of these interactive questions today ready to go, but technology said, So I'll try to do that next time. Okay, so here's what happens. Picture the neurotransmitter binding and letting sodium in. This is an action potential graph. We're going to be all over this in the nervous system chapter 2. But what's happening is, it said in the negative 90 in the slide before, but it could be negative 70 to negative 90. Let's put a little right here in your notes. Let's write RMP. That is resting membrane potential. Maybe I can do it. Okay, so this is RMP. We're hanging out, nothing's happening. Now, when the muscle gets stimulated, right here, this is right where we're going to say the neurotransmitter just attached to the receptor and sodium starts to go in. Sodium channels open. You saw that in that little animation. Neurotransmitter, the orange and yellow, binds to the receptor, and it's a ligand gated channel, right? Because it only opens when a chemical binds to it. As opposed to the calcium one before was a voltage gated channel. It opened because a action potential passed by. This one, the chemical binds, it opens, sodium goes in. Okay? Sodium goes in, and what happens to the charge inside the cell? It goes from negative all the way up. It passes 0, goes to almost like positive 30. It becomes positive. I mean, look how this is milliseconds here, very quick. Right when it gets towards the top of this, when it passes 0, so like in this, oh, by the way, a very important word right here. Moving in this direction, I'm going to draw an arrow, okay? Where we go from resting negative to positive is called depolarization. We're depolarizing that membrane because it's like, oh, it was so negative and positive out here, now they're coming closer together because we're moving to 0. So we call that depolarization. As we cross 0 and now it's positive inside the cell, what happens here is this. That doesn't work. The sodium channels are closing and potassium channels are opening right towards the top of this peak. So sodium channels close and potassium channels open. So no more sodium, we call it sodium influx or sodium entry first. Now that stops. Now potassium exits, positive charge is leaving now, instead of coming in. Now we go back to negative. That is called repolarization. So it's a quick depolarization, quick repolarization. Boom, that's a spike. Really, really quick because sodium going in and then potassium coming out right afterwards. So it's a quick flip-flop in charge. That is an impulse. That's an electrical impulse, an action potential, nerve impulse. That's how it works. When we get back down to about resting membrane potential, the potassium channels close and we just, little sodium potassium pump stuff to get us back to that resting membrane potential so we're ready for the next stimulus. That is an action potential spike. So right here is where the neurotransmitter binds to the receptor, and then it lets sodium in, and then right afterwards it closes and lets potassium out. And we get the spike. And then that spike is going to trigger another one right next door, right next door, right next door, and it's going to just spread across the whole sarcolemma, the surface of that entire muscle. Down those T-tubules. Okay, that's the next part.
Question.
Yeah.
You said to get those sodium that's already in, get them out. How do you get them out?
Well, okay, so the sodium comes in here,
Yeah.
right?
Yeah.
And then it closes, the sodium channels close, and then potassium is going to escape
Yeah.
really quick. And then what happens between the next stimulus, sodium potassium pump is doing its thing, getting rid of sodium, bringing potassium back in, kind of getting it set up back again for the next stimulus. Okay, so that's kind of what happens in, and I would write those steps down, like here, 1 through 5, or I sometimes spread it out even a little bit more. What time we got here? Just gonna maybe do this with you. Okay, we got 15 minutes. But okay, I'll do this one. I'm just trying to think. I might have a slide of this now, so I can save myself a little time. Let me see. There are slides. I think I put them a little bit. Let me just do this. Maybe not. Or I'm just not seeing it. Okay, I thought it was right here, maybe not. Okay, but you can write them down, and it's in the book, it just does it step by step, but it basically is everything we just talked about. Action potential gets to the, let's review. Okay, action potential gets here, calcium channels open, voltage gated calcium comes in, exocytosis of the neurotransmitter ACH binds to these receptors, which are ligand gated cation channels, sodium and potassium. First sodium comes in, up, potassium goes out. Okay, brings us back down, and then it's going to spread. So look at our next picture here. Okay, so this all happened right here. That happened. We caused an action potential right here at the junctional fold, at the motor end plate. Now it starts to spread. Picture it spreading in all directions. Now we've entered the next phase, the coupling phase. Excitation is just what happens right here. Now we're spreading, coupling, because now is that little latent period between stimulation and when the muscle actually contracts. So what happens? The action potential is generated here. It's going to spread. It's going to go down these T-tubules. Remember those? Little infoldings of the sarcolemma, cell membrane, so that the action potential doesn't just go along the surface to the end of the muscle. It's going to go deep inside. And then remember, next to the T-tubule on either side, we have these enlarged areas of the SR, the sarcoplasmic reticulum, called the terminal cisterna. Okay, the terminal cisterna are right next to the T-tubule, and as the action potential that's now coming down, we watched this video last time, it's going to cause, again, we have voltage-gated calcium channels in here. Here's calcium part 2. Calcium is stored in the SR and these terminal cisterna of the SR. When the action potential passes, calcium gets released everywhere in the muscle, all over the muscle. Okay, here's another image of that. So we had the excitation happened right here, first action potential, spreading down a T-tubule. Here's the terminal cisternae of that sarcoplasmic reticulum network, and it's going to cause those channels to open. Watch that video again. It's a great video of this happening in slow motion. Shows you every step of the way. Okay, the voltage-gated calcium channels open in the T-tubules, causing calcium gates to open in the SR. Same thing. I'm just putting pictures of the same thing. Here's the action potential down a T-tubule. There's the voltage-gated channels. Calcium comes from the terminal cisternae of the SR. Calcium gets released everywhere. Okay, this is showing the next part. the lock and key little analogy we talked about last time. Calcium is the key here, okay? Calcium is going to bind, we have our two contractile proteins. What are the two contractile proteins of muscle? Actin and myosin. Then we have what we call two regulatory proteins, troponin and tropomyosin. They're the lock, right? Tropomyosin is the lock, troponin is the keyhole. Write that down if you don't have it from last time. Just put that right next to tropomyosin. It's the lock. And troponin is the keyhole. Tropomyosin is a protein that is blocking these myosin binding sites, also called active sites. Myosin binding sites. Myosin wants to bind to these sites on actin so that they can pull and generate tension. But when the muscle is resting and not being stimulated, the tropomyosin is covering it. So they can't grab on. But when this happens, this action potential has come down. It's caused calcium to get released everywhere. Calcium is going to bind to the keyhole. Calcium is the key, right down in the corner. Calcium is the key. Calcium binds to troponin, the keyhole. That moves tropomyosin. It changes its shape. It moves and reveals the binding sites. So now the myosin binding sites, also called active sites, myosin can now bind to actin. And then we're entering into the next phase, contraction. Okay? Remember, this is all happening lightning fast. But all these things have to happen before that myosin can grab. And then we're going to get into our next phase of contraction. The lock and key. There's also a video of this. So, make sure you watch that. And we'll do this next time. I'll slow down a little bit, I promise. We'll do some videos. I'll ask you some questions because I know this is a lot. And this is really where the visual stuff is helpful. I find this to be really helpful. That interactive physiology, I can't recommend that enough. I just think you should take some time next couple days, just watch those, do those little practice exercises, and think of this whole sequence. But also, you're sort of zooming out a little bit and saying, okay, this is everything that, every time I contract my muscle, this is happening in my body. And knowing this sequence, you'll start to see, for example, I was going to ask a question about this, but there's you ever see movies that shooting like the indigenous people, like a little dart with poison on it? Yeah. There's a very common one that they use for hunting. It's called Curare. And what Curare does, it's a plant extract. It comes from a plant. And in certain doses, they concentrate it, put it on the arrowhead tip. This chemical binds to ACH receptors. It binds to them and blocks them. It prevents acetylcholine from binding to them. So that paralyzes the muscle. And think about the diaphragm. In an animal they're hunting, their diaphragm muscle is now not working. They're going to suffocate. They're going to die. It works pretty quickly. You don't want to get it with one of those. So we learn about all these different parts and seeing like some medications or some disease might block those calcium channels. So we can't even release the neurotransmitter. That's how some other neurotoxins can work. Botulism, things like that. You learn about how these different things affect different parts of these processes. So that's why it's important to understand these. Okay, so just a little preview because we may or may not have time. We're going to get to this next phase, the contraction phase. So now the active sites are open, the myosin heads. You see that remember the myosin had those little hooks, those little heads sticking out, so we're just zooming in on one of them here. And what's going to happen is, let's see if I can, because this really lends itself to animation here. But, throw this over here. So let's say here's a myosin, here's a big thick myosin thing, and we'll have one of these little heads sticking out. It looks kind of like that. Okay, like a little hook sticking out like that. Now there's lots of them all along the myosin filament sticking out. What's going to happen is, what they do is there's a little enzyme on here, and we have little ATP. They always draw ATP like this, like a little energy molecule. It's inside a little lightning thing like that, ATP. Okay, when ATP binds to this myosin head, there's an enzyme on it, it breaks ATP, okay, into ADP, right? Adenosine diphosphate, and there's the other phosphate. That was the triphosphate, but it breaks them up. So we have, let me just see if I have better, yeah. Okay, so ATP gets broken down to, there's a little, enzyme, myosin ATPase. Remember enzymes end in ASE? So it tells you what it does. On myosin, it breaks the ATP to ADP and an inorganic phosphate. That's what the ion is, just phosphate. And there's energy release. Remember, in ATP, those phosphate bonds have a lot of energy. So the energy gets liberated, and what do we do with that energy? The energy causes this myosin head to move into like a cocked back position. It like cocks it back. It's like spring loading it. Spring loads it back there. And then what it does is it's going to bind, let's say we have our little actin molecule here. It looks like these little actins, and it's got a little binding site. That's going to allow it now to bind. When it's cocked back, it now can bind to the active site, the myosin binding site. Then the next step is going to show it releases the phosphate and the ADP, and it springs back really quick. It springs back to where it was. But now, so here's the actin, here's the myosin head. First ATP comes along, it breaks the ATP, and it cocks back like this. It binds to the actin now. Now it releases the ADP and the phosphate, and it springs back to where it was. And it pulls the actin along with it. Then another ATP comes, it releases, cocks back again, grabs onto the next piece, pulls it. Okay? So I wish I could show this to you right now. Technical difficulties, but there it is. Okay, it cocked back. Now it binds. Now we call the power stroke. Okay, the power stroke is where, let me just get all the words up there. The power stroke is where it releases the ADP and the phosphate and bloop.
Can you put the reference?
Oh, sorry. Thank you. Okay, once it's bind, then it releases. So here it's going this way. 12 should point that way than there. Yeah. So the ADP and the phosphate release. Now it snaps back to where it was. Pulled it along. It's going to bind another ATP, which releases it. It's going to cock back again and maybe join to the next one.
Which part of that is power stroke? When it pulls back?
Yeah, the power stroke is when it snaps back.
Okay.
Yeah. It's pulling the actin along with it. And this is going to happen on both sides. They're pulling towards the middle of the sarcomere. I mentioned here that when you look at the myosin heads, let's see if I have a better picture of it here. It's really like two myosin heads together in each little thing. And one of them is grabbing on at any time. It's cocking back. And when it pulls, the other one cocks back. Because think about it, if all the myosin heads pulled at the same time, and then all released at the same time, what would happen? It would spring back and you wouldn't get anywhere. So one of the heads, it's kind of like you're pulling a rope, right? One hand pulls, if that one lets go, the other one pulls, so you don't lose tension, right? You're trying to pull someone like up a cliff on a rope, you're like, not gonna get anywhere, all right? You gotta switch, so they switch off like that. This is just a little sequence. Let's see where we start here. Okay, so let's just say, you can start anywhere. Let's say an ATP comes along, break it, cocks the head back here. Okay, cocking of the myosin head. Now, when that happens, it can bind to the actin, releases the ADP and the phosphate, and it power stroke. Okay, binds to another ATP, releases. That is why, we're going to pick this up next time. That is why after death, when you're not making any more ATP, some of these myosin heads are already cocked back when you died, and they actually can still attach to the actin and pull, and the power stroke can happen. That doesn't require new ATP, but you do need new ATP to pull the head off. So it stays contracted. Your muscles can stay tight after death, rigor mortis. And it can last for it can last a couple days actually, depending on temperature and all those things. So, you know, we need ATP not only to contract the muscle, we also need it to relax the muscle. ATP is needed to pull the myosin heads off so it gets ready for the next one. Okay? So that's the power stroke, that's the contraction phase. I know I kind of went through it pretty quick and there was a lot of steps there. We'll break it down a little bit, but do me a favor, do the study module, at least the part 1, chapter 9 study module. I can't show it to you now because I'm not online. But I'll send out a little reminder. I think that'll just really get you in a better position for next week to finish up this, next class to finish up this chapter. Okay? So